Hi everyone, and welcome to the webinar titled A Guide to Sequence Similarity Search for Biomolecules. My name is Nandana Madhusudanan, and I am from the web production team at the EMBL-EBI. So in this webinar, we will see an overview of sequence similarity search, why is this useful, and some basic concepts in alignment. And then the different sequence similarity search tools that we provide at EMBL-EBI and how to access them. We will then see how to perform a BLAST search for finding homologous sequences, and finally, how to navigate the result pages. Sequence similarity search is a method that helps you to find similar database sequences for your sequence of interest. Suppose you have a new protein sequence with a known function and you are interested to assign a function to this particular protein. How will you proceed? Well, you can make use of sequence similarity search tools where it aligns your sequence of interest, which is the query sequence, with the sequences that are already in the databases. And if you get a significant similarity among your query sequence and database sequence, you can consider them as homologous. That means they share a common ancestry. And the homologous sequence usually have the same or very similar functions. Likewise, you can find similar genes, domains, classify proteins, and explore evolutionary relationships using the sequence similarity search. Before we move on, there are some basic concepts that you need to understand. They are sequence identity, sequence similarity, and sequence homology. Sequence identity is the presence of identical residues between your query and subject sequences, whereas sequence similarity is the presence of residues with similar physiochemical properties. Here, you can see the residues arginine and histidine and histidine, and they are identical residues, whereas the residues arginine and histidine are considered as similar residues as they both are positively charged amino acids and have similar physiochemical properties. Sequence homology is the presence of ancestral relationship between sequences, and this can be inferred from similarity. Now we will see how to perform a basic sequence alignment. A sequence alignment is a way of arranging sequences to identify the regions of similarity, and it is the basics of database similarity search. Sequence alignment is easy if we have an exact match between sequences, but sequence is variable between individual species, locations, etc. Here is an example where we are trying to align a query sequence with sequence 1 and then with sequence 2. As you can see, we have two alignments here for the query sequence, alignment 1 with sequence 1 and alignment 2 with sequence 2. In this first alignment, we have 6 matching residues and 2 mismatched residues, whereas in the second alignment, we have 5 matches, 2 mismatches, and 1 gap, which is the site without a residue. A gap is introduced in the second residue, in the second alignment, in order to get maximum residue correspondence, and it represents the insertions and deletions of residues during evolution. The intel events occur very rarely in evolution, so gaps are always given a penalty. It is obvious that the matching residues are better than mismatches. Here given some values to match, mismatch, and gap in order to find the alignment score. An optimal alignment is the one with highest score. Another factor that we need to consider in alignment is the gap extension. It is considered that the adjacent gap positions are likely to have been created by the same intel event rather than multiple independent events. So we use smaller gap extension compared to gap opening penalty. When aligning protein sequence, we need to consider some other factors as well. That means how likely a residue is substituted by other amino acid residue. So there is this concept called a scoring matrix, which is a 20 by 20 matrix that are constructed based on the frequency of substitution and similarity of their properties. PAM and BLOSUM are the two main protein scoring matrices. PAM stands for point accepted mutation, whereas BLOSUM stands for block substitution matrices. So alignment score is the sum up of, for calculating the alignment score, we sum up all substitution scores from the scoring matrix table and subtract the gap penalties. There are two strategies for aligning sequences: global alignment and local alignment. Global alignment tries to find the best possible alignment across the entire length of two sequences. This method is more appropriate for aligning two closely related sequences with approximately same length. Global might not be suitable for sequences that are of a very different lengths or where there are sub-regions of interests. So there is this local alignment, which tries to find local regions with highest level of similarity. This is usually used when you need to find conserved patterns in DNA or protein sequence. There are different ways and algorithms to perform sequence alignment. We are not going to going through the details, but a dot plot creates a graphical representation of alignment by plotting dots where there is a match. In dynamic programming, it considers all possible alignments and take the best score. Mainly two alignments are used in dynamic programming, which is Dietelmann-Wunsch and Smith-Waterman. For local alignment, it is Smith-Waterman, and for global alignment, it is Dietelmann-Wunsch. For database searches, we need more efficient heuristic word method. Now, we will see the sequence similarity tools that we provide at the EMBL-EBI. For finding a tool, you can go directly to our service page, which is www.ebi.ac.uk/services. And this lists all our tools and data resources. If you know the tool name, you can directly search for it. Another way you can access the tool is going to the sequence similarity search tool page directly, which is www.ebi.ac.uk/tools/SSS. This is our menu page and it has links to other SSA tools. Since we have several tools, it is not always easy to choose the right tool for your analysis. Here are a few suggestions for choosing a tool. If you are interested in fastest search or only interested in highly similar sequences, you can use BLAST. On the other hand, if you needed a more accurate statistics for your search, you could use FastA suite of tools. If you have a short query sequence and you are looking for a global alignment in both query and database sequence, you can use GG search. If you have a short query sequence and you want to match all of your query sequence to a part of a larger database sequence, then you can use GL search, which is global in query and local in database sequence. For protein sequences, if you want to perform an iterative search, you can use the BLAST. It allows user to create a custom position-specific scoring matrix and which can help to find distant evolutionary relationships. On the other hand, if you need most accurate tool, you can use the search, which is also for finding distantly related protein sequences. Now we will see the tool input form. So here is an example of protein blast input form. All our sequence similarity search tools input form looks almost similar. So you can divide the tool input form mainly into 3 sections: database section, sequence section, and the parameters. We will see this in detail. As you know, we have a huge variety of databases available at EBI covering topics from genes, genomes, proteomics, metabolite system, etc. But for sequence similarity search, we only need the databases which has nucleotide or protein sequence information. Here, you can see the protein blast where you can search against protein databases. Whereas in nucleotide blast, you can search against nucleotide databases. You can select multiple databases and the number of databases that you selected will appear on the top here. The default databases for protein search is UniProt, KP, SwissProt, and for nucleotide search, it is ENA sequences. Now we will see how the sequence data is organized for sequence similarity search tool. First, we will see ENA, which is a huge nucleotide database. And the ENA database for sequence similarity search tool is mainly classified into a high-level organization. And this again is divided into data class and taxonomic divisions. As you can see, this is the high-level organization: ENA sequences, ENA coding sequences, ENA non-coding sequences, ribosomal, geospatial, and barcode sequences. A few of this high-level organization is again divided into taxonomic divisions and data classes. ENA data class is based on the type of data or the methodology used to obtain these data. These are the ENA data classes. And these are the taxonomic divisions in ENA. One thing that you need to keep in mind here is that there are some exclusion in the taxonomic divisions. Rodent excludes mouse, and mammal excludes human, mouse, and rodent. Vertebrates exclude human, mouse, rodent, and mammal. So these exclusions are only for sequence similarity search tools and not for the ENA browser. So if you want to search against mammals, you need to include human, mouse, and rodent separately. Also, suppose you selected the database ENA sequence mammal, and you are trying to filter the result for human taxonomy. You may not get the correct result as human is not included in mammal. These organizations allow you to search against a smaller data set. For example, if you are searching for a particular taxonomic division's EST, you can select it directly rather than searching the entire EST. Another major database we have for sequence similarity search tools is UniProt protein databases. If you are interested in high quality manually annotated and reviewed protein sequences, you can select the UniProt KB SwissProt. For automatically annotated and unreviewed protein sequences, you can select the UniProt KB Tremble. And there is UniProt KB SwissProt isoforms. For SARS-CoV-2 related protein sequences, there is UniProt KB COVID-19 database. There are taxonomic subsets of UniProt KB database as well, and UniRef databases are also available in sequence similarity search tools. Other databases include patent protein and patent nucleotide sequences, and sequences from structural databases like PDB-E, We also have some special databases like IPD, Intact, Kemball, etc. So the second section in the tool input form is the sequence input, where you can either paste your query sequence or use the file upload option. If you are searching with an entry which is already in our database, you can provide the database colon identifier. The tool will fetch the sequence data information for you. And the sequence that you are providing should be in a proper sequence format. As you know, different databases store data in different formats. You can provide your data in any of the sequence formats like EMBL, GenBank, PIR, etc. And the preferred sequence format is FASTA. Please note that the input sequence should be in a plain text file. Our tools won't support .doc files sequence formats. So here are some examples of sequence format. This is the EMBL sequence format, and this is the GenBank format for the same entry. And this is the FASTA format. So the next section in the tool input format is the parameters. Every tool has a set of default parameters like matrix, gap open, gap extension, etc. If you want to use a different value for any parameter, you can click on the more options and change it accordingly. Once you have the input and parameter set, you can click on the submit button. If you wish, you can provide your email address here, and the links to the result will be sent to your email. Once you submit the sequence, you will get a status page with the job ID. And once the search is done, you will be provided with the result page. Please note that the results are stored in our servers for only 7 days. So it is better to download the alignment if you need to access it later. Now we will see how to perform a BLAST search. So this is the URL for BLAST. And I'm going to use the query sequence spike glycoprotein, which is already available in the UniProt KB SwissProt, and I am taking the sequence in FASTA format. So this is the result page. This is the input form for NCBI BLAST, ProteinNCBI BLAST, where you are trying to search your query sequence against protein databases. I'm using the default parameters here. So I got the job ID. It may take a few seconds to load the result. So in theory, I will be getting the same entry as the first hit because I took the entry which is already there in the SwissProt database. So as you can see, this is the result page, and it has a result, different tabs to different result pages. And these are the hits that I got. And you got 50 hits here. By default, the result page shows maximum 50 hits because it is said the default value for the alignment number of hits is 50. If you want to change, you can change that in the parameters. Now let me go back to the slide again to explain this in more detail. So these all are the result tabs. So this is the result page, and the first page is the summary table. So the first tab in the result page is the summary table. The first column in the summary table is the number of hits that you got for your result. The second column has the link to the database entry. The third column has a description about the entry and cross-reference. This will help you to get more detailed information about the entry from other databases. Next column is the length of the sequences, the hits. And then we have the alignment score and percentage of identical and similar residues, and then E-value. So we are more interested in the scores and E-value of the alignment. Ideally, we need a high score and low E-value for our alignment. Now we will see what is an E-value. E-value is the measure of chance of an alignment occurring randomly. That means how likely am I going to be wrong in assessing homology as a result of that hit. E-values are represented like, for example, 1e minus 3 in our result page, which means 1 into 10 to the power minus 3. That is happy to be wrong in 1 time in 1,000 times. The E-value depend on this database size and the length of the query sequence. So short sequences are more likely to occur by chance, so may have higher E-value. And similarly, if the database size is big, then more possibility to find alignment by chance. So you should be careful while analyzing your result. On the left-hand side of the summary table, we have a selection box, which allows you to perform several actions on selected hits. It has options to show database annotations, show the alignment between query and database sequence, and option to download the database entries either in FASTA or EMBL format, and also have options to pass your entries to other multiple sequence alignment tools. Second tab in the result page is the tool output, where you can see the actual alignment. Here, you have an option to download the alignment. It has the information about the database name, number of sequences in the database, etc. On this, if you want to go to a particular alignment, you can click on that alignment. So in this figure, you can see the alignments for first and last hit. Next tab in the result page is the visual output. Here you can see the region of query and database sequence where the alignment is found. This will help you to check if the alignment and the annotation you are looking for is at the same part of the sequence or not. You can change the color scheme to dynamic or static. Dynamic is based on the range of E-values in your result, and in fixed, it is based on a fixed range that is 0 to 100. Next tab is the functional predictions that shows the domains and family predictions of the hits from UniProt. On the left-hand side, you can see the UniProt databases. These are the regions that are matching the UniProt databases. You can make use of this information to make sure that you are not aligning domains that are unrelated to your protein. Next tab in the result summary. Next tab is the result summary tab. You can download the tool results from here. Links to different results formats are available in here. The final result tab is the submission details that has all your submission details like input sequence, parameters, databases, submission date, etc. It has It also has the tool's version information. There are some tips that may be useful for you while performing your sequence search. It is better to search the smallest database that is likely to contain the sequence of interest. You can use the default parameters for most of your queries, but if needed, consider different parameters as well. For finding homologous sequences, use E-values rather than percentage of identity or percentage of similarity. It is always better to look at the other informations, especially domains, descriptions, and annotations. As you know, homology can be inferred from significant similarity, but if they are paralogous sequences, they have different functions even though there is a sequence similarity. So further work might be needed for your analysis. If you are interested in finding relatedness of your sequence data, try to perform multiple sequence alignment among your hits. Multiple sequence alignment tools are available from our sequence similarity tools result page. If you want to learn more about our tools, you can have a look at our documentation page. And for any queries related to our services, you can contact us directly at this mail ID, support@ebi.ac.uk. Thank you.